Hello, dear students. Today we will continue with the blood cell formation. We already studied red blood cells and white blood cells. So we are left with the platelets. That's the third category of formed elements found in blood. So let's study the platelets now. Platelets are again formed from the stem cells in the red bone marrow of any person. These platelets are basically responsible for formation of a blood clot during an injury when bleeding is taking place. So let's study the platelets now. The platelets are also called as thrombocytes. These platelets are called as thrombocytes because they are cells which are basically involved in formation of a blood clot. The word thrombus means a clot. So the platelets are oval or round in shape. They are non-nucleated and they are membrane-bound structures. They have a thin membrane around them. These are about 2 lakh to 4 lakh per cubic mm of blood. Their concentration is higher, much higher than that of the WBCs, but lower than the red blood cells. They have a lifespan of about 3 to 5 days. So after 5 days, the platelets die. They are destroyed mainly in spleen. So once their death has to take place, the platelets, old worn out platelets, are removed and destroyed in the spleen. Now, another thing about platelets is that they are formed from a cell which is very huge in size. So they are budded off from a giant cell called megakaryocyte. The red bone marrow has the stem cells which undergo various developmental stages to form a structure called as a megakaryocyte. This megakaryocyte will form a bit of its nucleus budding off with the membrane, and that structure is called as a platelet. So the platelet is basically budded off from the giant megakaryocytes. So this is how the megakaryocyte looks like. The megakaryocyte is a huge cell, much bigger in size than either the red blood cells or the white blood cells. And the platelets are budded off by having a little bit of its nucleus surrounded by a thin membrane. The platelets basically help in formation of a blood clot. And what is a blood clot? When we get injured and there is loss of blood from the point where an injury or a wound is formed, we have all noticed that after some time that injured blood thickens and ultimately stops flowing. This is because there is formation of a blood clot over the point of injury. This blood clot is essential to prevent excessive loss of blood. And this is the property of, or the function of, the platelets. So let's study the formation of a blood clot. Now, what are the sequence of protective steps taken by the body when a bleeding or an injury takes place? So first thing, when we have an injury or a bleeding blood vessel, the first thing your body does is it constricts the blood vessel. It reduces the diameter of the blood vessel so that the blood flow to this injured area can be reduced. Then after the blood flow has been reduced, a temporary plug of platelets is formed. The platelets in the adjoining area, they become activated, or we say they are injured. They clump together to form a temporary plug, preventing any loss of blood from inside. Once a temporary plug has been formed, in between, the platelets will start a step of reactions to form a fibrin mesh. This fibrin mesh will block the opening externally. Okay, so it will branched network of fibers which is formed, interconnected network of fibers, and this network of fibers will now attach onto the same platelets and RBCs which try to escape out, plugging this opening, and this is called as a permanent blood clot. So we have these steps taken to prevent loss of blood from an injury. So how is the blood clotting taking place? How is that the platelets change something into the fibrin mesh? So the various steps are: the injured platelets, they change and they form a chemical called, they release a chemical compound called the thrombokinase, It is also called as thromboplastin. These days, instead of just calling it as thrombokinase or thromboplastin, we call it as factor X or Stewart factor. This formation requires the presence of calcium ions. So in the presence of calcium ions, injured platelets will form Stewart factor or factor X. Now, once that has happened, the formation of factor X has taken place, then the prothrombin, which is the inactive form of a protein present in the blood plasma, will change into thrombin, which is its active form. Now, once thrombin has been formed, has been activated, then this activation again requires the presence of calcium ions and vitamin K. Now, here the vitamin required in blood clotting is K. So in the presence of both of these, calcium and vitamin K, the prothrombin is activated to thrombin in the presence of factor X. Now, once thrombin has been formed, it will change the soluble protein fibrinogen into the insoluble fibrin. Now, again, this synthesis requires the presence of calcium ions and vitamin K. Once fibrin insoluble has been formed, it will form a network or a mesh by having interconnected fibrin fibers. We call it as a fibrin polymer. It's a network of sticky threads, as you can see here, a network of sticky threads. Once this network of sticky threads has been formed, it will plug the opening of the injury, and any oozing out RBCs or platelets stick to it. This will be now called as a clot. So a clot or a thrombus is formed when the injured platelets release thromboplastin. So by release of thromboplastin or thrombokinase or factor X or Stewart factor, the entire sequence of events is brought about to lead to formation of a clot. This clot will now prevent any loss of blood by the site which has got injured. Now, normally we say that injury happens externally, that injury happens when the body, we fall down or you injure yourself externally. That means you're exposing your body to the external environment. But sometimes a clot formation may take place while the blood is not exposed to the external environment. One such type is called as arteriosclerosis. Here a clot may be formed inside an artery. All of us have arteries which have a diameter which is quite wide. The arteries can expand, they can contract, they carry blood from the heart to the other organs. So a healthy artery of a person will have a big diameter to allow a good flow of blood. But sometimes, due to accumulation of fat, calcium, cholesterol, or other such substances which harden over time, we say that the inner lining has become slightly misshapen. Here, the inner lining has the development of a structure called plaque. So plaque is basically made up of fat, cholesterol, calcium, other substances which are formed in the blood. And this plaque constricts the opening of the artery. The artery is narrowed. Now the blood will have to exert pressure in order to move across this narrow opening. In exerting that pressure, the blood vessel may be damaged, the plaque may ooze out, and there may be injury which takes place to the platelets. So the platelets may become activated, and on activation, they may lead to formation of a clot at the site where the plaque has oozed out or where the opening had become too narrow for the blood to pass through. So there is a formation of a clot inside the artery. This is called as arteriosclerosis. Arteriosclerosis, as you can see here, in this case, the blood clot is formed inside the arteries. Okay, the blood, this is the inner lining of the arteries. A blood clot has been formed here on the inner lining due to deposition of plaque. Now, if the clot remains here, that also is not good. But if the clot moves along with this and blocks some narrow artery elsewhere, That is also not good. So anywhere inside the body, if a clot is formed, that clot needs to be removed by medication or some kind of a surgical treatment. But the clot formation inside the arteries is not good. This is called as arteriosclerosis. That means clot can be formed without exposure to the external environment. The blood can clot in a test tube. When you take blood, a sample of a blood, and you allow it to stay, or you put a twig or a piece of wood to stir it and you let it rest for some time, a clot can be formed at the bottom. Red blood cells, white blood cells, and platelets at the bottom, and the blood plasma remains above, because the density of the plasma is much less as compared to the density of the formed elements. Or a blood clot may form under skin. This is when somebody bumps or hits against a surface. So a bluish or a purplish patch of skin is formed, indicating that the blood vessels just underneath the skin got ruptured. The clot was formed while the skin was not exposed to the external environment. Or you may have a blood blister. And what is a blood blister? A blood vessel may rupture under the skin. Maybe a part of your skin was squeezed or pressed in any object, and that skin portion swelled up. And along with this, the blood which was here clotted underneath. So all these three conditions indicate that clotting of blood does not require exposure to the external environment. It only requires the activation of the platelets. Once the platelets have formed the Stewart factor or factor X, the blood clotting will take place. Now, there are two defects due to improper clotting of blood. The first defect is called as hemophilia. Now, in case of hemophilia, blood loss keeps on taking place. Even from a minor injury, the blood does not clot, or it clots improperly. This is basically due to absence of clotting factors in blood. It is a hereditary disease. That means it is inherited. It runs in families, and it is on genes, located on genes of an individual. So it is one disease due to improper clotting of blood. The second disease is viral dengue fever. Here, the number of platelets fall due to an abnormally low level, which often, so hence the blood does not clot, or sometimes the blood may settle down in certain places. Coagulation, that means clotting of blood, is very slow. So it leads to excessive blood loss inside the body also. That's called as hemorrhage. It's caused by the bite of Aedes mosquito. So one person infected, if that person's infected blood is sucked by Aedes mosquito and the Aedes mosquito bites another person, transmits the viral dengue fever. So this is also due to improper clotting of blood. So both of these defects are due to improper clotting of blood. So with this, we finish the function and the role of platelets, various types of clotting. And now we will be doing the process of blood groups and how they are required during blood transfusion in the next video. So hopefully you have understood all this about platelets in this video. But make sure you revise this topic. Thank you very much, and God bless you all.